think all the people that I met with earlier today, I've really had a great day. Everybody I met with actually was doing relevant work to what I'm interested in, and doing it in great depth. So actually this talk I won't have a lot of great depth because I wanted to give kind of a more broad overview and kind of focus on sort of the big questions. So kind of apologize a bit for not going into great detail in some, but in the question and answer period we can pursue some of those a bit more. So this is what the brain looks like if we were to take it out of the skull, and the MRI looks at the brain like this. So the MRI sees boxes of white or gray. The boxes are about a milliliter. You can get them a bit smaller with more powerful magnets and more time in the scanner. But most of the literature, it's these boxes classified either gray or white. And within each of these boxes then would be millions of neurons and maybe trillions of synaptic connections. And this is simultaneously my most condescending and profound slide, in the sense of that it's kind of a cautionary note because the images can be very colorful and the movies sort of irrationally persuasive sometimes. But to realize that it's sort of still a crude measure. It's kind of looking at a city from an airplane. We can see kind of the major shopping centers and roads and stuff, but we're not getting down to the kitchen room tables kind of thing where we really want to be for some of the synaptic activity. So as a cautionary note to myself and to you in terms of the limitations of the imaging technology. And a lot of the challenges for the next decade are trying to look deeper inside of these boxes, which is being done here very well in terms of really, I think the key is going to be we're going to have to look at non-human primate studies to be able to look under microscopes to really kind of look at the relationship between the colorful MRI images and what's really happening kind of under the hood and inside of those boxes. And so actually my first contact with Dr. Amaral and stuff were around this kind of work of really looking at the link between the basic kind of science and development and across species leading up to humans. So this, I think, is still a very active and exciting road for the future and to interpret the images. So the gray and white, what is mostly gray in terms of shown here is in turquoise. The cell bodies and where the DNA resides, the connecting points on the axons. It's sort of nuanced, though, in terms of what we call gray matter voxels, even though this is for a mouse, it's probably quite similar for a lot of humans. So even within something that sort of gray matter is cortex, you can see that there's a lot of nuances in terms of it's not just synaptic connections. A lot of things go into what we call gray matter, and a lot into what we call white matter, which is shown here sort of as kind of I kind of think they look like little hot dogs. The little white wrappings along here are what the MRI sees as white matter. So our study design is sort of non-creative in a sense. I mean, the basic idea is we have kids come to our lab as young as we can, which is for us is about 3, and then we do brain scans, we do DNA collection, and different types of testing for cognition, behavior, emotion, and then we have them come back and just try to follow how these things change both in health and in illness. And so we have just over like 6,000 scans now, and half of them are healthy, happy, typically developing kids, and half of those are identical or non-identical twins. So that's sort of the basic idea in terms of it's kind of data gathering in terms of as opposed to hypothesis driven. We really want just to see how does the brain grow and how does it change from babies to children to adolescents to adults. And this kind of 1, 2, 3 approach, the first mapping part took about 13 of those 19 years. It's pretty much exclusively just creating the map. How does it go? And then we switched to this second objective into, well, what matters, for good or ill? What nudges these paths of development? Does anything that we do as a parent or a teacher or clinician matter? So this is when we became very interested in twins, nature-nurture type of questions. All of this, eventually, we hope, will get us to the third of being useful, guiding interventions. And so some of the translational work I heard about earlier here, today and stuff, really doing some great stuff, toward that end. But it's been kind of frustrating in terms of how sort of really tangible things that we've gotten from imaging and probably genetics as well, in terms of what do I do different now with all this imaging and genetics as a parent? What do I do different as a person, as a society? And we haven't really delivered much on that. And of course, still trying, and a lot of people, I think, we haven't given up, but it has been daunting in terms of, even after cracking the genetic code and all of these things, in terms of really making things useful on a personal level, it's been challenging. So what I was going to do now is just sort of break down what we've seen over the years in terms of how white matter does change and how gray matter does change through the years. And what we see from white matter is that it increases from not just the first 2 decades of life, probably the 5th decade for women and the 4th for men, but as long as people are reasonably healthy and don't have demyelinating things like MS and stuff, you get more and more white matter as you go through development. And it's not subtle in terms of, so if you get these hot dog things, this insulation that wraps around, it can increase the speed 100-fold versus non-myelin. It's like 200 miles per hour for the fastest connections. And so that's not very subtle. And it's not just a matter of how fast things can go. So when you have this insulation, and it is actually kind of insulation functionally as well, just like insulation in wires, but the electrical signal kind of hops in between the nodes of the hot dog. And so the actual ions going across the membrane and stuff only have to happen at these nodes. And so it's much easier to reload in terms of, instead of having to move all the ions back across the other side, which takes energy and time, you only have to sort of reload at these nodes, and that gives you the ability to fire again 30 times faster. And so the combination of those is a pretty impressive, like 100 times 30, a 3,000-fold increase in bandwidth or ability to move information. And so that is definitely not subtle. It's not uniquely human, but to this degree, it's very distinctive to our species. And I think underlies a lot of the behavioral changes and clinical symptoms that I'm going to talk about next. It's not completely random, the parts of the brain that are necessary, like for life support. We need to breathe and our heart to beat and those kind of things myelinate quite early. Then it's the 5 senses in terms of sight, sound, touch. I always forget one, smell, taste. But the so-called primary sensory and motor. And then it's the things that tie those together. So the association areas that tie together. It looks like a duck, sounds like a duck, those kind of things. And then that keeps going in terms of associating the association areas. So there's some kind of rhyme or reason to the sequence of development, but mostly it's ever onward. And when we started these studies, we naively were thinking we were just going to watch the brain grow. Your brain's this big, now you can do arithmetic. Now it got bigger, now algebra. Bigger is still calculus. That we would see these straightforward, bigger is better size relationships. So it was really kind of disappointing. We thought that by 6 years old, first grade, the brain is already 93% of adult size. So we're like, oh no, this is going to be very boring. It'd be like watching grass grow, 7% difference over 2 decades. Like it's totally be uninteresting. So then it's still actually that's true. I mean, the total size of the brain does hit close to adult pretty early, but the different parts within it undergo dynamic changes. So it's like the frame or the structure of the house is built, but the wiring and rearranging the furniture and stuff goes on. So very kind of slow changes in general, but if we look at real local levels, so this is the corpus callosum, which is the connection between the left and right half of the brain. And so we can see that in certain parts, in just like a 1-year period, an 80% thickening of that part of the corpus callosum. So at local levels, things can be surprisingly dynamic and changing quite rapidly. And so this has generated a lot of interest in technologies to get a better handle on these white matter changes. The diffusion tensor imaging, again, great work being done here on that. Magnetization transfer imaging, a lot of sort of jargon and long terms, but just another way to try to capture these white matter changes. One of the recognitions from this is that it's not just a matter of sort of pedal to the metal, let's move signals as fast as we can in the brain, the faster the better. It's that it's more a matter of the fine-tuning of the speed that's important. So if you want to have the front part of your brain integrating information from the visual system, which is in the very back, and the auditory system, which is on the side, you actually don't want the signals to be going both at the same speed, because they have to arrive at the same time. And so you actually want the visual to be going faster than the auditory, and it's exquisite timing, like 50 milliseconds. The input has to arrive, and then the neuron adds up all the yays and all the nays and decides, whether to fire. But it's really, really fine-tuned. And the white matter helps to really guide that in terms of getting it just right. The white matter, as it is growing more and more, also then inhibits more axonal sprouting, in a sense, decreases the ability for plasticity and change, which may have a lot of implications as well. It's sort of part of the deal in terms of the trade-off. You have these sort of fewer but faster connections. So growing up in a fairly rural community in North Dakota, an analogy that comes to mind for me is you have gravel roads. They're very nice grid laid out. Every quarter of a mile you can turn off to a perpendicular road. Then the roads that get used a lot might become paved. The roads that get used even more between major grain centers become multi-lane highways and stuff. Now you can zoom down them, but you can't turn off every quarter of a mile anymore. And so that's the trade-off in terms of the connections that you use and get heavily myelinated can be much more efficient, but fewer choices along the way. So this also raises this idea of sensitive or critical periods that I'll talk a bit about. But the real key, I think, is that these white matter changes allow the brain to communicate within itself better. So one of the ideas when we first started, we were going to look at different parts of the brain. We say, well, hippocampus does memory. Prefrontal cortex controls impulses, and which people don't probably mean as literally, but that's not really how it works. They're all different parts of the brain are parts of networks or whole groups of structures. And it's probably better to think of the different brain parts as like letters of the alphabet. And so the prefrontal cortex is involved in lots of different functions. That may be like a vowel. That may be the letter A, or the hippocampus, an E. But what happens in development isn't so much then developing new letters. It's that those letters get combined into words, and then those words into sentences, and the sentences into paragraphs. And so the real developmental change and maturation is in the combining itself. And so that's sort of the white matter changes, fMRI, resting state, graph theories. All these things are trying to get at this idea of how the brain talks to itself in terms of the different components, and whether that's the key to developmental change. This is for all the British neurosurgeons in the audience. They're doing the operation. There's a doctor, what's the matter? Gray from the looks of it. And the matter with gray matter, so to speak, is that it doesn't act like white matter, which isn't at all obvious, right? From conception through death, they're constantly interacting. They're part of the same neural circuits. They should be maturing quite alike, but they don't. So we have the white matter is increasing across all the regions. Again, if we look really locally, there is some specificity, but frontal, parietal, temporal, it's all going up. And gray matter has an upside-down U path. And if we think of that upside-down U, then the peak of it, the top of it, could be looked at as one indication of maturation, and that's very different by region. So it's not that the top of the U, I think, is the peak ability part, but at least it's something we can quantify, and it can give you a sense of early versus late. And so they're quite different in their paths. And the peak for frontal gray matter is about 11 in girls and about 13.5 in boys. And that actually, for us, is kind of a big difference, like 2.5 years. And we thought, oh, kind of run to the educators and like, guess what? The girl's brain is maturing faster than the boys. And the teacher's like, duh, we've only known that for 100 years now. Thanks for nothing. You can go back to your lab. But for us, it was at least interesting that we saw this male-female difference in their trajectories. And so that became sort of a theme in terms of trying to capture these trajectories. So the upside-down U isn't everywhere in the brain. This is relatively recent that we looked at this, that it's only the parts of the brain that are, in a sense, sort of more recent evolutionarily in terms of the 6-layer cortex. And so they can be 3, 4, 5, 6. So the more primitive in the sense of like reptiles and things having, so the 3-layer cortex they don't change very much with age. The intermediate ones are linear, they increase with age. And then the more advanced sort of 6-layer cortex, that's the one that follows the upside-down U. So we kind of have this loose link between the more complicated the structure, the more complicated the growth curve. And not to push that too far, but we thought it was an interesting sort of one of these ontogeny, phylogeny things that true evolutionists hate that kind of simplistic notion, but it kind of fits in that pattern of what do we see in evolutionary history and individual history. So what's causing the gray matter to change in size? Unresolved in terms of, are we actually seeing this idea of arborization, so arbor, the Latin word for tree, growing extra twigs, branches, and then pruning. Is it more brain cells? Is it blood vessels? Other things. And so that's one of the key unanswered questions in terms of what's behind the MRI changes. Very early in life, there's data in terms of what you see here. This sort of they get bushier, extra branches, twigs, and roots. A little bit of stuff from adolescence sort of suggesting that, but it really needs to be nailed down a bit more about what's actually creating the changes. So this sort of 1-2 punch, you overproduce and then you fight it out. Some neural Darwinism, sometimes sort of survival of fittest connections, and it's a very commonly used approach in nature, and actually in the physical universe in biology. That's how almost all complexity arises in physical systems. Overproduction and non-random selection. You have some sort of competition. Really, it's the heart and soul of evolution, I guess, that same notion. So we have seen it for the gray matter changes, but it's not just gray matter. So post-mortem data is consistent with this. EEG changes, particularly in coherence, MEG, PET scans. This is very often cited data from the wonderful work of Peter Huttenlocher showing this childhood buildup, decrease, and leveling off in adulthood, except there's no real data for the adult. There's like 4 data points in this entire world lifetime work, which is the best there is as well. But it's really not resolved from a post-mortem perspective. This is for dopamine receptor systems, but also some data for norepinephrine, serotonin, acetylcholine. This general notion, with plenty of exceptions, but this childhood build-up, fierce competition during adolescence, and then leveling off during adulthood.
The brain grows like a tree. First, there is a flurry of growth. Then unused branches or pathways are pruned. And it is this pruning that gives the tree its shape
So this
for
is,
the
again,
future.
not to be like condescending, but I thought this was really neat in terms of, this graphics and cartoons by do you guys get the Zitz cartoon comic strip out here? But it's sort of focused, on adolescence. And we've used this to sort of reach out to the students themselves and stuff in terms of regarding, brain plasticity. And I think it's very important and not done nearly enough in terms of going directly to the schools and students, because they, as a group, they like this stuff, and I think it's empowering to know that, yeah, what I do can change how my brain grows and what it specializes in. And so partly for that, partly sort of to put my plug in of the need to disseminate these kind of informations and to have more interactions both ways, the neurosciences with the educators and the other way as well. So I said like the top of the U is different in different places, and so this is maybe it takes a little while to kind of get a handle on, but the bluing can be looked at as maturation, in the sense it's getting closer to the adult levels. And one area that is notably late to turn blue is here in the dorsolateral prefrontal cortex, and it's, again, it doesn't do impulsivity, but it's a prominent part of the circuitry involved in long-range planning, impulse control, organization, those kind of skills. And it doesn't really hit the adult levels till age 25. So that seemed like ridiculously late to us. Some adolescent psychiatrists, okay, we're starting this study. Should we go to age 16 or 18? Right? By 16, they're already kind of big. They're getting adult dosage of medicines for a lot of things. People then, and still argue now, it's only since the Industrial Revolution that we kind of created this whole notion of adolescence, and that in most biological ways, they're already done. So we're like, well, we'll overshoot, we'll play it safe, we'll go to 18. 25 wasn't even on our radar screen in terms of as a difference. And so we kind of joke the only people who got this right were the car rental companies, because if you go to the airport and you rent a car, you have to be 25. So, no wonder Hertz is all the best neuroscientists. That's the problem you're dealing with. But it didn't go away. In terms of, they have tons of data in terms of that 24-year-olds cost them more than 54-year-olds, which isn't like, that obvious, right? Their reflexes are better, their vision's better, everything you measure like on a simulator and stuff, they do better. They're not novice drivers. In the U.S., they might be driving for 6, 7 years. And this kind of led to other, this notion of the paradox of adolescence. You should be at the top of your game, tolerant to heat and cold, resistant to cancer. Almost everything you measure, you should be, at your peak. Morbidity and mortality go up 200, 300%. Number one, and by far, is car accidents. So if you're a teenage girl and you die, half the time it's motor vehicle accidents. I have a 17-year-old daughter who started driving. It's staggering. And nothing else is close in mortality statistics. In terms of prostate cancer, men over 7, maybe 8, you start getting in high single digits. But that's sort of staggering. 2 and 3 are suicide or homicide, depending on how people classify the events. But all the top 10 seem to be related to judgment, decision-making, emotion. So this movie doesn't, prove that, or anything, close to that. So I want to be careful not to overplay, this relationship. But it's more as a generator of questions in terms of, is there a relationship between these parts of the brain maturing relatively late? And it's not that they're gone, right? It's better to think in terms of not as good as they're going to get. It's not, that it's absent. But that's become a focus for a lot of our questions and things in terms of if this part of the brain, which also is involved in kind of integrating past, present, future, what is all of our memories, our knowledge, what's happening in the here and now, where are we, what is the input, what are our hopes and dreams for the future, sort of being able to play out scenarios in our mind instead of in the physical world, which is pretty adaptive for dangerous outcomes. So it's really kind of a very critical part of the brain, quite different between us and non-human primates. And if it is relatively late to mature, so what? And so what might be the implications of that for our society, for the judicial system. This is actually used in the Simmons-Roper Supreme Court case to raise the death penalty from 16 to 18, razor-thin 5-4 decision. And I'm not saying it should have been used, but sort of it is, in terms of should people be allowed to get birth control when they're 16 without their parental consent. So it kind of gets, I think, extended into very complex, nuanced social and public policy things. But the neuroscience, if we want to ignore them, it's at our peril because it's already out there. And so how to kind of use but not abuse this type of data is a big challenge. What does it mean for a parent if our children aren't quote as good as they're going to get in terms of just because they're going off to college? Do we butt in more? Do we, what does that change what we do? Because in so many of our big decisions we have to do before 25, marriage, career, military service, well, just a bunch of things. We can't wait that long. And the other kind of, I guess, phase 2 of my research. So what does matter? I mean, does anything we do as a parent? What about medicines for ADHD or autism or schizophrenia, depression? What about diet? Video games, which is huge in terms of the amount of interface time with electronics is exploded. Even from 10 years ago, it's very different. And all these things are sort of like good or bad. I mean, I'm not sure, but it's very different in terms of the social networking and Facebook and just texting and stuff. It's a huge part of most middle school and high school people's lives. Does use it or lose it matter? Is that sort of the driving principle in terms of how the brain optimizes? And if we have this 1-2 punch, you overproduce and you fight it out, what if something tilts the battlefield of that? What if something goes different? Does that lead to ADHD, dyslexia, learning disabilities, other sorts of things? This has just opened back home where I am in terms of the Hall of Human Origins. And one of the things that I thought was interesting from that exhibit was looking at the relationship between brain volume and environment. And before seeing this, I was more thinking like, as times get tough, then our brains get bigger because we have to sort of be more clever to survive in those environments. And that when droughts or bad things happen, then we'd have to have kind of brutal pruning. But the ones that survived, then they would have the bigger brains moving forward. And this is sort of subtle, but it's not so much the difficulty of the environment that looks to have driven that, it's the change itself. And so the punchline from this exhibit was sort of that it's not so much survival of the fittest, but survival of the versatile. That what's really distinctive about humans is that we're very versatile. We can adapt to different environments. Longer than other animals, we have this vulnerable but long period where we're kind of dependent upon our parents. But we are incredibly resourceful in terms of humans. We can live on the North Pole. We can live on the equator. We can even live in outer space with the technologies we've developed, everything in between. It's 10,000 years ago, hunting and gathering berries. Now we're doing computers and PowerPoint, these things, entirely different kind of activities. And that's not very long. 10,000 years in evolutionary terms, that's a blink of an eye. Totally different day-to-day stuff. I thought of like reading. Most humans have never read a single word. So reading's about 5,000, maybe 5,200 years old. And since splitting from the other hominids 6 million years ago, there's been 100 billion humans. So most humans that live and died, reading wasn't even invented yet. And yet, a lot of people in this room, a lot of our waking hours, we spend reading. So the incredible thing is just it's so changeable, so plastic, and so that's I guess the blessing and the curse, how to harness this in terms of this brain's incredible ability to adapt to the environments we present it. So what should we present the environment? That's what school is in a sense. We're creating an environment that we think the brain will adapt to. How do we know what will be most useful for the adults in their world? How do we use this information for good. So the theme that comes from this idea of yes, my entire career in a fortune cookie, very humbling, but it's neuroimaging, like life itself, is about this journey, not just the destination. And one way this came out was looking at this gray matter changes, the cortical thickness. How does that predict IQ? And if we look at age 19, so blue is the high IQ group. So these are all typically developing kids, over 125, so very, very high IQ group. Green is in the middle, 110 to 125, and red is average. And if we just look at the gray matter thickness, no luck here in terms of we can't really tell anything at age 19. It's only when we run the clock backwards, when we look at the path of how it got there, that we see the separation. And at least to me, this is somewhat surprising that actually the later peak was the high IQ group. And that was, I think, kind of interesting to wonder about. It raised a lot of questions, more questions and answers about these like tortoise and hare kind of things. Is it better to do things as early as possible, or not necessarily? And are there people who may develop slower but longer and ultimately achieve as much as more? This got some interesting sort of press things when it came out. Like LA Times sort of looked, bigger is better. New York Times like smaller is better. The pinnacle of my career, I made capitalist imperialist pig. I almost retired that. Well, I can only go downhill from here, but I didn't have anything else to do, so I kept going. But it's kind of frustrating because everyone kind of missed the message. It wasn't is bigger better or is smaller better in terms of. You can see that, here smaller is better, here no difference, here bigger is better. It sort of was bittersweet because it invalidated the prior 10 years of a lot of our work in terms of this kind of focus on, is bigger better or smaller better or that. That's sort of like the wrong question in the sense where that's not, without looking at age, all bets are off. So this is one of the sort of themes that we've been pursuing in terms of the trajectory. And then it also raises this idea of critical or sensitive periods, because when the brain is very busy, when there's like lots of arborization and pruning back down, like for the visual system, for vestibular system, for these things, that sort of reflects as time of sensitive periods. So can we work backwards? Can we identify these times when the brain is really changing a lot? Again, in the crude sort of MRI sense, we're looking at millions of neurons at once, but can we work backwards and find these sensitive periods where things are happening a lot? So one approach or a line of investigation is looking at the basal ganglia, which I've been talking mostly about, the outer surface of the brain, the cortex, but the basal ganglia, they include the caudate, shown here. Some people think it looks like a dolphin leaping out of the water. The globus pallidus putamen. These are like more inside the brain, and so the pictures generated by Paul Thompson at UCLA in terms of just I just thought it was aesthetically kind of interesting to see in terms of quantifying these things, and their sort of window, if you were, would be between the ages of 7 and 11. So it's where we have this huge increase in the number of BET cells, or these kind of huge brain cells involved in fine motor control. So we see a 50% tissue loss of the BET cells in this window. And so this would sort of suggest, again, this is kind of speculative stuff, but this is like a really important time for the environment to have an impact. So if you talk to coaches or music teachers, they usually endorse this, if you want to have a world-class musician or be really good at something, that you should be doing it before 11. 100% of the Atlanta Olympic gold medalists were doing this by 8. Or if you bring, your daughter to a figure skating coach and say, she's 12, but she's a gifted athlete. She was, a great soccer player. All these things, they'd probably say no. He's like, no, she's coordinated. No, it's like too late. In terms of to be, world champion, it's just, could you get really good, have a great time, pick up golf in your 40s? And yeah, all of those things. But to be, at the extreme levels, that there could be these notions of sensitive periods. And the basal ganglia that I showed earlier, they're interesting from an evolutionary standpoint. The way this is often compared, you convert everything to the same size of 70-kilogram hedgehog. I'm not sure why hedgehog, but that's what's usually used. And now let's compare the different brain parts. What's most different between human and hedgehog is the neocortex. So that's 156 times larger in humans than the hedgehog. But in second place is the basal ganglia, distant second, but the only other thing in double digits. And so that what this kind of implies is that this increase in basal ganglia size occurred relatively recently in our human history, and this sort of notion then of moving parts gets broken in the sense of the things most recently acquired, less genetic redundancy. And in the literature, almost, if you had to bet on a multiple choice test in terms of, for almost every pediatric neuropsych thing, guess the basal ganglia will be different. So this is just to summarize what we have seen in terms of white matter goes up, gray matter upside down U, relatively late for the prefrontal cortex, which I did talk a bit about, didn't talk as much about superior temporal or inferior parietal, but they as well are very late to reach adult size. All of them are very, quote, high association areas. They tie together information from all around the brain. That we need to look at not just the destination, but the journey, the path of how it got there. And the new, I guess, front is wondering about the notion of critical or sensitive periods and whether that might be important for education or clinical interventions. So that was anatomy of the brain, and as many people in this room know far better than I, the MRI can also look at brain activity or physiology, blood flow. And so what you see from, in general, in these studies is this changing balance from frontal to limbic. This is a study that's been around for a while, not done by us, but Deborah Yergel and Todd's group at Harvard. So you show a picture of a face, and the teen has to say, is this person surprised, afraid, or sad? And when you have the teenagers and adults do this, then one, the teens don't do as well, but the adults will use more of that sort of late developing dorsal prefrontal cortex, whereas the adolescents are more likely to use the amygdala, hippocampal, or the limbic sort of things. So not to overplay this one study too much, but it's sort of a general theme that you have this changing balance between more frontal involvement, less limbic across a lot of tasks. Another kind of general theme, which with plenty of exceptions, but in general, you see that the activation gets more and more focused, more and more precise from childhood to adolescence to adulthood. And you get more integration of different parts of the brain all going up and down together on these tasks. So those are sort of kind of three themes that are actually pretty consistent across the anatomical and the functionals, that the brain is growing, it's becoming more integrated, more connected, more specialized in a sense, at the cost maybe of being plastic and able to learn all new things. You kind of have to choose in a sense of what the brain is going to be optimized for. So part two, or the switch then, is so that's what we see. Now what matters? So what affects these things? Twin studies are a natural sort of option to wonder about nature and nurture questions. So we've been trying to look at identical twins and not so identical using this thing called structural equation modeling, which, again, not to get too jargony, but the A is what people would usually consider to be like heritability in terms of common environment, unique environment. And what we see, probably not so surprisingly, is that is the brain heritable? Depends on what part. So different parts of the brain are heritable to different degrees. The outlier in these, so most of the different brain parts we've looked at have about the same kind of range of 0.8 or so heritability. The cerebellum sticks out as a weirdo in terms of it's the least heritable of the structures we've examined. So if you look at that corpus callosum again, the connection between the left and right hemispheres, They're so alike in identical twins that complete novices could come up to a panel of 20 images and say, these two are a pair, these two are a pair, these two are a pair. Cerebellum, that'd be really tough. They're not that much more alike in identical than non-identical. So in addition to being the least heritable, it's the most different between boys and girls, and it's the latest to reach adult volume. So it's got a lot of like features that make it interesting from a pediatric neuroimaging perspective. So this is the development of blue in boys and red in girls, so about 15% difference. It's even more than the difference in total brain size. The real sort of twist on this for us, though, was that the answer also changes a lot by age. And actually, we hadn't anticipated that. In terms of designing the study, we went to the twin experts and said, how many twins are we going to need? And they were like, we've got good news for you in terms of essentially doing imaging, and you can kind of quantify it and be precise and stuff. You probably wouldn't need very many at all, about 10,000 should be fine. And we're like, wait, sort of, excuse me, kind of thing. It's like, for imaging, that's actually, a lot. And so we had to kind of, aggressively bargain with them. So like, well, we won't answer your calls until you talk about at least 1,000 and stuff. So we kind of went back and forth in terms of what would be a minimum amount to get meaningful data. And all of that, though, didn't really anticipate that there would be these age by heritability changes. So it's not even is the prefrontal cortex heritable or not, it's at what age. And so this has been another focus for us in terms of how does heritability change with age? Is it because certain genes become active that weren't at birth? Is it because of epigenetic factors? So at the beginning, I would have thought that identical twins will drift apart every day they're alive. The environment is not identical, even if your parents dress you the same and you have the same haircut and stuff. Every day's going to be a little bit different. So as the time goes by, you're going to get a little bit farther, but not so. So both in terms of imaging stuff, diploma and stuff, in terms of a lot of psychology, you actually get more alike during adolescence or other times. So that was at first very puzzling, less so now when we consider that not all genes are active at all times in life. But so this has really become a focus for us in terms of if there are genes, if certain parts of the brain are becoming more alike during adolescence, are they also related to why illnesses emerge during adolescence? Is there a link between heritability? It also has implications for study design. So if we look at gray matter heritability in general, that goes down with age. If we look at white matter heritability, that in general goes up with age. So you can kind of think of the gray matter and the synapses and stuff maybe responding to the environment more. Again, I wouldn't want to have to defend that too vigorously, but sort of that's how I kind of think of it. White matter is sort of on autopilot. It's mostly going to increase in volume. And then kind of all bets off for the cerebellum. It has this only thing that really shows this increasing unique environment effect quite late in life. The other part was multivariate analysis. And what this does then, it's like not just is it heritable, but are things driven by the same genetic or environmental factors. And this surprised the twin people that a single genetic factor accounted for 60% of cortical thickness variability. So at first I thought, wow, what gene is it? So it's actually not necessarily a single gene. So it's more of a mathematical idea of a principal component. But it would suggest actually that it is, if it is a lot of genes, they're not independent. So it'd be like one pathway. But that sort of was surprising to them that it might have been something like a doubling, one more doubling step or something that is a fairly abrupt increase in human cortical thickness from apes and stuff, that it could have been sort of not just a little bit bigger because of language development or fire use, that something happened that made a big difference. And then when you take that factor out, that 6 factors accounted for over half the remaining variants. So these numbers are actually quite a bit lower than we thought, where you have like schizophrenia, like 1,000 genes each contributing a very small effect. And so this is the other side of that coin, where as a relatively small number of factors may have pretty widespread global effects. The other big theme is boy-girl differences. It's hard to escape in terms of everything that we do as a child psychiatrist, different ages of onset, symptoms, prevalences between boys and girls. And so we wonder whether these differences in typical development relate to clinical. But without really thinking through, I mean, it's a very politically sensitive, topic. There's been horrible, things that have happened from discrimination and have happened, are happening, as we speak in terms of educational access, discrimination. So we really wanted to be, careful with this, but it's kind of unavoidable. I mean, so many of the genes that we look at are different between boys and girls. And consistent with this theme, what we find, the total brain volume is different, but what is even more compellingly different is the trajectories. So that almost all the trajectories that we've looked at, I think 8 to 18 months earlier in the females. So we have questions like, is the corpus callosum different between boys and girls? This is like one of the most studied sectomorphin things, 160 papers. Now, about a third say it's bigger, a third say it's smaller, a third say it's not different. And it all depends, like, do you correct for a 10% difference in total brain size? And even, it also depends on, like, at what age. So 4 to 7, yes. 7 to 11, no. 11 to 14, yes again. And so a lot of things we've looked at follow this pattern. It's like at what age are you talking about? Because the answer changes, and they crisscross as they go along. So that was another sort of trajectory, not destination difference. What's most strikingly different is that the variability is greater in boys. So almost all the highs and lows are different, are the boys. And I just confirmed something I was wondering about before when I was talking to Tony Simon earlier in terms of things like math disability is actually more common in boys, as are extreme math high scores on SATs and stuff. But so there might be some kind of clue here just in terms of like the differences in variability related to boys having the XY and the girls with XX. There may be sort of some averaging out type of effect. But this is, everything that's not white is where the variance is higher in boys than girls. So it's pretty much everything. And so we're wondering, is this hormones? Is it X and Y chromosomes? Is it society differences? So one of the ways to try to sort this out is to look at boys and girls with extra X or Y chromosomes and boys and girls with high or low hormonal differences. So Klinefelter syndrome extra X chromosome is the most common of these, and what we found was a wide range of abilities and academic challenges, but it's kind of a consistent story when we looked at the parts of the brain that were different, the temporal lobes on both sides. So this would be also areas like involved in language and social things, the frontal areas that would overlap with what you'd see in ADHD, the motor strip, and also an interesting negative. So the parietal part of the brain involved in spatial manipulation things is not only preserved in XXY, but it's actually enhanced, or it's more extreme. So you could have sort of you could have a different pattern of academic strengths and weaknesses. The other aspect of this work has then to look at not just XXY, but like dosage effects. So have people with 1 to 5 X's and 0 to 2 to Y's to see each extra X or Y you get, you tend to have more clinical problems, which is not so obvious for the X's because the extra X is inactivated. Each extra X you get, but not completely. So about 85% of it is. But that sort of narrows the search. Then, if you're getting worse clinically with each extra X, then it should be linked to that 15% that's not inactivated. This was a surprisingly consistent pattern in terms of probably don't have time to look at it closely, but the XX, so that would be like a typical healthy female. If you add an X, the brain gets smaller. If you add a Y, it gets bigger. And it holds, it's completely consistent. So if you kind of follow it along, every time you add an extra X, total brain size gets smaller. Add an extra Y, it gets bigger. Again, bigger is not better in terms of it really isn't. I mean, it's not just sort of dodging this issue, but I mean, I'm not saying, that part, but it could be something on the X and Y's that are related to total brain size. And other differences are more related to specific parts. We've been interested in this androgen receptor gene, because whether you're a boy or a girl, if you have the different types of this gene, which relate to how testosterone works, it also predicts, just as you would expect in terms of the differences in men with high and low androgen receptor activity, looks like differences between men and women on a lot of the imaging and behavioral variables. This congenital adrenal hyperplasia is just an example of one of the group that they have XX or XY chromosomes, but their hormone levels are quite different. And what we see here is that it affects the amygdala volume, but not total brain size. So just we're trying to sort of sort out, like, what are these differences related to hormones, what are related to X and Y things, not hormone, what's related to society and other things. So we're looking at a lot of different groups that have different levels. So just to kind of wrap up the sex difference things, so overwhelmingly more alike than different. So if you show me a scan, I can't say this is a boy or a girl. But what is most different is the trajectories. The male brains are more variable, but these are group differences, right? And there's lots of exceptions to the rules. So an example I think that is appropriate is that if you consider height. So that's actually different between boys and girls. 100 men randomly selected, 100 women randomly selected. Almost every time the average height of the men will be higher than the average height of the women. But if you were to go into a school and say, okay, everybody above 5'6", this locker room, everybody below 5'6", that locker room, people would be outraged. It's like, why? Well, there's too many exceptions to the rule. There's so many women taller than so many men that would. But that actually is like twice the effect size of any of the imaging or psychological things in terms of, so if you were to go into a school and say all boys should be taught this way, all girls that way, it'd be even sort of more ridiculous than height alone. So that's the kind of subtlety of trying to convey group averages versus individuals. So I'm just going to wrap up a few notions, specific genes. This is, we paid $500,000 for the egg of a supermodel and the sperm of a Nobel laureate. She didn't turn out quite like we planned, which is kind of like happens to us in terms of it's very hard to know whether genes are good or bad, so to speak. It depends on the age and the context. This is looking at the, quote, bad Alzheimer gene in kids, and we actually see the same brain imaging changes based on whether you have the good or bad Alzheimer genes, but absolutely no impairment in terms of they don't do any worse psychologically. But that was, we were wondering about whether it affects the brain. Illnesses, and I have to wrap up here, so I'll have to refer people to the papers on this, but this general notion of why do things happen in adolescence? If you're kind of born with the genes or the risk, so many things, not everything, not autism, not ADHD, not Alzheimer's, but lots of other things emerge during adolescence. And so we've been wondering whether the normal, typical changes in adolescence, if they kind of run amok, if they go too far, is that related to illnesses? And sorry for flashing through these, but just to establish this point, for schizophrenia, it's kind of yes. All the changes you see in schizophrenia would be predicted if you went the normal changes, but too far. This is the movie showing that upside down use. So gray matter is supposed to get thinner during adolescence, but in schizophrenia, it's fourfold. Again, so that's like not very subtle. So it's 4% in typical, 28% in schizophrenia. That doesn't mean this is what's causing schizophrenia, because they already had it when they entered the study. But that's one of the kind of things we're wondering about in terms of this whole idea about pruning, overproduction, selective elimination. Is it a factor in these things? And ADHD, looking at, is the brain different? Again, we did see some snapshot and time differences, but more compellingly, it's when we look at the trajectory of growth, where we see this kind of delay frontal maturation in ADHD. So this is the concluding slide, and I'm sorry if I keep harping on this one point, but I think that's the key sort of summary of our work in terms of that. If we consider the journey, not just the destination, it predicts the cognitive and IQ and behavioral differences. It predicts male-female differences better. It has a big impact on genes and environment, and health and illness. So this is my, if you only take away one message from this would be it. So thank you so much for your attention, and I don't know if I have any time left, but I do if you guys do. But thank you very much.
Sorry about that. We're going to take questions. There's plenty of time for questions, so I'll try to trot a microphone in over to anyone who wants to ask.
That was a wonderful talk. Thank you so much for joining us. You know, we talked earlier about variability, and in my lab, that's one of our focal points. Do you think, as we discover more about, you know, the complexity of the brain, that a lot of what we keep trying to look at in terms of differences is it's going to be, we think, much more complex. So it's not just going to be age, it's going to be, you know, environment, it's going to be genes, there's going to be many things. And so as we go through our data now and looking at these things, do you have any strong suggestions of what we should be considering, not just gender, not just age, so that we can try and, with our current data, start uncovering some of the complexity?
I think the variability itself, in terms of where there's so much individual variation. I didn't show one of the slides, but just looking at total brain size for two 10-year-old boys, both doing great, and they're double the difference in brain size. So there's a huge sort of wiggle room. And a lot of it initially was like how to get rid of this variance, and kind of variance was bad. And now we see it as our friend, you know, in terms of the variance itself, as what causes it, why are different people different, and to really try to go after the variance itself in terms of you know, what it relates to. But in addition to sort of this notion of needing to wonder about the trajectory, so bigger, smaller, tying that back to at what age and boy or girl, and trying to look at some of the differences being explained by those things. But the other, I think, big paradigm shift has been this notion of small world networks, graph theory, different kind of terms for getting at networks. So how do we capture the connection between all the different parts? And so it is hard. I mean, I think it's looking back, we'll kind of almost feel foolish for trying so hard to find this chunk of the brain predicting, social, very complicated, incredibly, nuanced things of getting along with other human beings and stuff. We'll say, why do they even think that, that would be predicted from, a big chunk? But it's both kind of like a blessing and a curse. But the variability is, it's always striking, even if you kind of know that it's going to be a lot, that boys and girls, doing well or doing similarly can have such different findings in biology. Tony?
So, given all these beautiful results you've shown about cortical changes with this long extended pattern, and the secondary very big effect, although much smaller, from the basal ganglia. When you have congenital disorders, does that maybe point a little more to the basal ganglia as being sort of the root of some of these atypical developmental trajectories? Because presumably there's a lot more established, so there's a lot more potential impact for things having gone wrong early in those.
Even especially so for the basal ganglia, because they're one of the few parts of the brain that doesn't have a backup blood supply. So they're very vulnerable to shearing effects during the birth process itself, and having only that one sort of source of blood. So they do show up a lot in terms of when, I should have showed the slides I have of identical twins. One of them has ADHD and one of them doesn't. And when we see that, very often we're seeing more of a birth kind of trauma to the basal ganglia. And so it sort of almost makes it even rarer than it might think in terms of to find two identical twins. One definitely has ADHD, one doesn't. When that happens, often it's the affected twin does have sort of like a reason to have it. And so in ADHD type of stuff, if you have damage to the basal ganglia, then you often have symptoms of ADHD or frontal lobe or cerebellum. And so you almost need all the things to not be damaged, for it to not have the illnesses. And that's a challenge as well, because any kind of link of the chain that's broken could lead to the final common pathway. And certainly, all these autism, we're wondering, like, that there's a lot of things that could lead to that picture. But the basal ganglia, as a cruel joke by some, are harder to measure. They're the cortex and stuff is pretty automated and stuff now, and a lot of the basal ganglia stuff is sitting in a dark room like a medieval monk, outlining again. So that's been part of the story in terms of doing that. But it needs to be done. I mean, I think in terms of what the basal ganglia do, long list the more you look, but they're so connected to the frontal lobe activity and the cerebellum. And so that's been another kind of theme in terms of how to not do these, I can't remember what the metaphor is like, but the lampshade where you're looking for your keys because that's where the light is. There's a lot of ignoring of the basal ganglia for that, and the cerebellum as well. It's actually kind of a real challenge to measure. But I think that's going to be kind of moving forward a really important part of the circuitry.
Hello. Make a long story short, when I was 6 years old, I drowned, which erased my memory and ability to feel fear, sorrow, apathy, facial expressions. Prior to that, I had a very little bit of affection, which I showed towards animals and things, according to my mother. But throughout my life, I've had a very powerful level of imagination, which is kind of like associated with the journey and not the destination.
Yeah.
And so that was part of what I didn't hear anything on imagination, because that's all I've had my whole life, was a very high level of imagination. And not so I won't take up so much time, but my psychologist said that I don't know how you did it, but somehow you've been able to rewire your brain. Because today I can read facial expressions, I can read somebody touching me. So many different things affects me in ways that I never knew existed.
Yeah.
So throughout my life, my imagination has empowered me to be able to overcome many challenges and get to where I am today. And I realize, I think something in up to around age 4 or 5, we have a very high level of imagination, but somewhere it's after 5, maybe 6, it starts to dwindle away, and by adolescence, the imagination is really like not there. And so I think, because of my ability to look at everything and imagine away and just not accept it for what it was, to have that open mind has enabled me to be able to stay on this journey to where I am today at 49, and I have a mind that feels like it's quite young. So
Yeah.
I think imagination may play a very big key here in what, this journey's all about, and getting the brain to act in the way it does.
Yeah. Well, one, I'm very glad you're with us, in terms of that is good. And it actually touches upon a lot of these powerful themes in terms of the plasticity, the brain's ability, to change and adapt, even when there have been trauma or environmental things, sometimes very extreme, in terms of at different ages, the outcomes might, be different. But that even, like, people can have literally half of their brain sort of removed because of this thing called von Rasmussen's encephalopathy things, and remarkably sort of degree of function returned in terms of honor students graduating from sports teams and stuff. This notion of what the brain can rewire in the sense. And so things like that, people say, well, how could that person without half a brain, how could they even have language? Because language is in the left, it's right here in this spot. But that's not really how it works. Like for the cerebellum, which is in the very back, we kept trying to think, well, what does it do? It helps you not tip over in a bike. It's involved in coordination and balance. And then the next paper, no, it's involved in matrix algebra. And the list just grows on. And so it became apparent what the cerebellum does is a certain type of math. It does this sort of parallel processing. It happens to work well for tipping over in a bike or for these other things. And what do language centers do? It's not language, it's a certain type of math. The different letters, I guess, as I said before as an analogy, but the different parts of the brain do a certain type of math or information processing for language. Usually that works well for that left hemisphere part, but it's not the only way. It doesn't have to be. And so for your brain, if certain parts were damaged at certain times when the brain was still very plastic, things like imagination, we usually can think of that would be the frontal lobe stuff. And so maybe your frontal lobe had to work harder than it might have because of other parts being damaged. But then a kind of good outcome of that, then by really drawing upon that part of the brain that is involved in imagining, because a part of imagination is this kind of time travel notion. It's like pretending, like, well, what if? And you can kind of run out the scenarios in your mind instead of the real world. And maybe by kind of using that part of the brain more, that yours was strengthened more than it would have been if it didn't have to work so hard on those other things. So there's lots of, I think, very encouraging examples like yours where people were able, with persistence and things, to arrive at very good outcomes because of the brain's changeability and plasticity. So, I think that's the good side of it, and that, as for you, that there's a lot of potential for the brain to overcome obstacles.
Thank you.
Hello. I had a question. I'm a school psychologist, and I was wondering, it may be a very general question, but as a school psychologist whose primary role is to do an assessment to determine eligibility for special education and different learning styles and disabilities, what would be the take-home points for me to keep in mind from your presentation in terms of determining if a disability exists, doing screeners for ADHD, those types of things?
I think the take-home message is mostly very upbeat, in terms of that our harnessing of the plasticity in the brain. We are just, I think, scratching the surface in terms of that one, that even people who look to be have gone down the wrong path, for quite a time, biologically the ability to change and to acquire new skills and stuff is there, for a long, long time through this, I think forever in terms of, but there are sort of trade-offs where as the plasticity isn't as great as it is at other ages, but this idea in terms of the brain being able to change itself in response to the demands. The other thing, I think, education-wise, I didn't talk about this as well, as how powerful modeling is in terms of the brain sort of learning by example. And it's really, unlike reading, which is sort of unnatural, in terms of there's no brain center. But what the brain is very good at is learning by what peers and parents and stuff are doing. And so I think that's sort of unrecognized of how, as parents and teachers and stuff, we're not just teaching when we sit down and have these somber talks and stuff. It's all the little everyday things of how we treat our spouse, our fellow people, and how we handle stress, all these little things I think are really having a powerful impact on these developing brains, and it's a little unnerving or scary, in terms of that responsibility we have. But I think that's what you're doing is something that's for me the most interesting next part about how do we understand education. How can we use the neuroscience advances to help education even a little bit? If we improve it 1%, millions or maybe hundreds of millions of kids a little bit better. I mean, the leveraging is phenomenal. But I think that sometimes people think of, oh, this is a lost cause. This 10-year-old is already stealing cars. He's already doing, there's nothing we can do. And maybe socially that is the case. It would be very hard to get people out of gangs and stuff. But biologically, that's not the constraint. I mean, biologically, you can still turn things around dramatically.
From what this gentleman just said about drowning and how he kind of gained emotions, would you say? And a number of other things. Is there hope that autistic children will someday outgrow what they're going through right now?
I think so. Yeah, I mean, that's a great question in terms of, and my sort of, yes, in terms of, I really think that this changeability is so powerful. Will it be exactly the same? Will they kind of use the same brain parts in the same way to get to the same end? I think probably not. And I don't want to be like sort of like Pollyannish, of like, oh, this is just a matter of trying hard enough, or so that for a lot of people that they will have impairments or they will have different strengths and weaknesses, throughout. I think we have a long way to go, but the essence sort of how plastic, how changeable the brain is, I think we have so much more upside that we haven't realized yet. And how to sort of, I guess, keep that in mind, but without sort of making it untrue, and sort of just tell people, no, don't worry, that any day now, people will come up with a medicine or a computer intervention that will, make everything better. I think that's not how it will work as well in terms of, I guess, understanding the in-betweens of what I just said in terms of, like, that's really going to be where the advances are going, to move forward. Because if we understand how it works and if we can really understand what is different in autism, then we can really get going, and then we can kind of see what we do as a parent and as a clinician or schools and stuff to really kind of understand it. That's going to, for me, that would be the place to start.
Hi, I was wondering if you could go back to the previous slide and kind of go over its implications in terms of with this population.
So this is, I kind of going through so fast that I probably should have been a little more cautious in sort of not suggesting that ADHD is, quote, just a delay, in terms of, because that's actually did sort of, get misconstrued that way in terms of we saw that the gray matter changes in terms of the frontal lobe maturation that was even extra late in the ADHD kids. That's where impulsivity and long-range planning and stuff, so it was almost like too tidy, in terms of oversimplifying it. Because I don't think you can really have just a delay in something, because the world doesn't stop around you. So if you have different impulses, it's not that a 10-year-old ADHD person is just like an 8-year-old person without. And that's how a lot of people kind of interpreted this, or sort of like almost like, don't worry, just wait 2 years, everything will catch up. And so that was kind of the downside to this. But the ADHD story is, initially I think it was looking for the, kind of like the hole in the brain, kind of thing, or where's the damage. And what I didn't, there isn't damage in that sense. It's not, there's no gliosis, there's that, I really do see it as like a difference as opposed to a defect. Not to say that it can't, be a problem or a challenge and stuff, but in that sense, I think it's really, wondering in terms of the limitations for people with ADHD. I would say they're not there. You can achieve things. It might have to be a different way, different path. And some people, they just, you can have other things. You can be brilliant or have mentor. That's not saying that, it's like a good thing to have in terms. But I think in terms of like brain-wise, that I was very struck by, like, we don't find more. The radiologists don't find more flat out, pathology and stuff. Almost exactly the same as, people without. But the idea then, if you're going to have like the delays, it's going to be harder to hold back impulses and be less distracted and stuff, then how do you kind of acknowledge that and work with it so you don't also like punish people for not doing things that that they can't do. The effects of medicines, we really don't know in terms of longer term effects, but they seem to help in the short term. And the other thing that we're just talking like a couple hours ago in terms of that if we look at people without ADHD, but that have different levels of impulsivity and stuff, it's almost like the same movie. So now I'm like more confused than ever in terms of like the idea, is it a long continuum? I used to think, well, kind of, but there's if you look at like a bell curve, there's two humps. Yeah, some people are in between, but there's definitely a group, that. And so now I'm not so sure, because imaging-wise, it looks like that even within people that don't have the diagnosis, that you'd see this pattern. So I'm kind of less certain than I was 19 years ago when, we started this. But I think like these other things, I mean, particularly that there isn't, pathology. There's no, autopsy, funny business, and that kind of thing. So if this is a difference in trajectory, then one idea is sort of to like prevent irreversible mistakes, or hard to reverse: motor vehicle accidents, incarceration, addiction, sexually transmitted diseases. There's sort of some like the kind of choosing your battles sort of thing, keeping educational options open. Maybe you'll take longer to get through the college, but so what? In terms of then, it doesn't mean that you won't get through. So the jury's still out, so to speak. But the overinterpretation of this was that don't worry, it's just a matter of catching up. Sorry for that was a very long-winded, diffuse answer, but.
The frontal lobe there is the part that's really standing out in terms of underdevelopment.
Yeah.
If I'm reading it right.
You are. So that's sort of the message in terms of that it's that part of the brain, is even later to mature in a sense, and that would be related to impulsivity, long-range planning, organization particularly. Yeah.
Other questions?
In regards to the sex difference in variability,
yes.
What does the developmental trajectory of that sex difference look like? Are there certain periods of increased variability?
Yeah.
In contrast, the sex difference.
Yeah. So I had some of these like movies on my laptop, but I moved them, for two of the different things. So I wish I could have shown this one as well. But the differences between men and women, or males and females over time, so they kind of look like a difference in what we just saw, like in those movies, in terms of the latest to sort of catch up is the dorsolateral prefrontal cortex. And so the movies of the differences between males and females, which wasn't obvious, that was very recent, just like last month when we kind of looked at the kind of maps of movies of difference. And so certain parts are more related to puberty itself in terms of amygdala, hippocampus, cerebellum, and other parts seem to be just age and not hormones. So we're trying to sort that out a bit. And the other thing is just huge variability, in terms of, and even behaviorally and stuff, like some 14 and 15-year-olds are, very mature, very non-impulsive and stuff, and some people never outgrow that. And in terms of that's a tough one in terms of like how does the judicial system and stuff deal with this idea that there's just huge individual variability, and from person to person.
Very brief curiosity. Your results about IQ and the different developmental trajectories, the different IQ, made me think that IQ levels also change across generations, and you have such a large data set that you may have already looked at cohort effects in developmental trajectories of the brain. I wonder if you have.
Yeah, we haven't yet. I mean, that'd be actually interesting to do. We kind of wondered about like if the peaks are later, because I didn't want to apply to like if you read when you're 3 or whatever, young, that's not like a bad thing, sort of. But the idea then is like, oh, then you're gifted, and then you get sort of whisked away to a whole different education thing. What about the person who didn't read at 3? I think the trouble is then a lot of those might be, do just as well, but you're kind of like culled pretty early in the process, and then from then on your whole educational experience could be different. And so math particularly, like if you the cut, like in third grade, how fast can you multiply and divide, and that's actually not even that math. Like, it's more of a verbal skill, it's more temporal, it correlates very highly with vocabulary ability. And so if you kind of then say, oh, you're a gifted math and you're not, you probably miss a lot of people who would do really well, at higher math later. But one of the speculative interpretations was, if we look back at like that sensitive period stuff, so at the time of these peaks, that's when sort of the most action is happening, and that in a sense then the high IQ group was at its most plastic time kind of later in life, and so in a sense it's adapting to a 12-year-old world instead of an 8-year-old world. And it's not sort of that's inherently sort of healthy or not, but it's sort of more adaptive. If you're kind of at your peak input from the environment in a more complicated thing, then that's like a good thing. Good in the sense of it helps you, with adult stuff. And we see, we've looked at like prodigies, people who at age 10 acquired adult skills, and where are they now kind of thing. And it's sort of a pretty consistent theme, and people do okay, but they're not, they don't sort of stay at the very extreme and stuff. There's a lot of examples of the tortoises, people who kind of slow and steady, but ended up going, keep improving longer. I think the imaging stuff doesn't directly address it, but I think education reform-wise, it's an interesting thing to wonder about. Is it always earlier better? It's like Tiger Woods after that phenomenon. It's like there's so many 3-year-olds now, doing golf. It's not going to work, I don't think. Even though he did that, but that's not the, it's not like the only thing you need to do to have, gifted athletes or musicians. And although there is that notion of 10,000, you can, you can sort of, I think, create genius in a sense, or at least highly skilled things. So a lot of this is, that it is, fun to wonder about how much expertise you could if you, started from birth. But educational system, it's huge in terms of trajectories, in terms of should we teach kids to read younger and younger? And so I just came from Scandinavia, where they're looking at comparing education across different countries, and some countries or school systems, they start reading at 4 or younger, and others at 7. They don't even try before. And by 12, there's no difference. So if you start reading, that's relatively late. Almost always people catch up. So then it was like, well, maybe 4-year-olds shouldn't be reading. Maybe they should be playing, in terms of, but playing is complicated. It's not like that's nothing, in terms of getting. And so I think a lot of these things are just fun, but we don't know. But certainly as a parent, it makes me wonder a lot in terms of, because I probably bought into that as well. Like, get them the right software, and you got to be crunching numbers. You're 4 now. You're free ride. No more. You're a freeloader. Get to work on these things, but maybe it's not what their brain is supposed to be doing at that age. They consider it a very sort of American sort of notion, like, younger, early better. But.
Okay, I think we've taxed you enough. Can you please join me in thanking Dr. Giedd for
Thank you very much. Great questions.
The UC Davis Mind Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.